Hello everyone, welcome back to my channel Cyanide Medicine, and today we are going to see that how ATP is being made in our body and what is electron transport chain. So before we start, subscribe to my channel Cyanide Medicine so you cannot miss my future video. So without further ado, let's get started. So as we all know that our body needs energy to work or to function, or that energy is in form of ATP, and that energy producing process called electron transport chain, which occurs in mitochondria from NADH and FADH2, which carries electron, and those electron passes through series of structures and make ATP, and that's how we will produce energy in our body in mitochondria. In this video, we're gonna see later on that how electron is being passed through different type of structure. But first, we will see what is aerobic metabolism. Aerobic metabolism means metabolism in the presence of oxygen. In aerobic metabolism, glucose will go into the glycolysis, and that glycolysis occurs in first cytosol and in the mitochondria. In cytosol, glucose will give 2 ATP, 2 pyruvate molecule and 2 NADH molecule, and those pyruvate molecule enters into mitochondria for glycolysis in TCA cycle. In mitochondria, that pyruvate molecule will enter into TCA cycle and will give 2 acetyl coenzyme A, and that acetyl coenzyme A will give 2 GTP, 6 NADH and 2 FADH2, and these all will make ATP. Remember that 2 NADH in cytosol is still outside. We have to get those 2 NADH into the mitochondria, and we are gonna see how those 2 NADH will get into the mitochondria in next slide. As I have mentioned before in my previous video that how NADH is being transferred into the mitochondria, here's I'm gonna explain it again. Oxaloacetate will convert into the malate. In this process, NADH plus H plus will convert into the NAD plus in cytosol, and that malate will go into the mitochondria, and that malate will convert back into the oxaloacetate, and it will free one NADH molecule, and that oxaloacetate we have to get it out, and that oxaloacetate will convert into the aspartate, and that aspartate will transferred out from special transporters outside into the cytosol, and that's how NADH is being transferred into the mitochondria. Apart from the malate shuttle, we have another shuttle which is called glycerol phosphate shuttle. In this shuttle, it will also transfer NADH2 into the mitochondria, but not exactly transferred, it will convert into the FADH2. Let's see how. In this shuttle, dihydroxyacetone phosphate will convert into glycerol 3-phosphate, and in this process, it will convert NADH into the NAD+, and that glycerol 3-phosphate will convert back into the dihydroxyacetone phosphate with the help of glycerol phosphate dehydrogenase enzyme, and that enzyme is embedded into the inner membrane of the mitochondria. Because of that, it will simultaneously convert FAD into the FADH2, and that's how we will get one FADH2 molecule inside into the mitochondria. So that's how NADH is converting into the FADH. So we are not exactly transferring NADH into the mitochondria, but we are converting NADH into the FADH2, and that's how FADH2 will go into the electron transport chain and will make ATP. So electron transport chain extracts electron from NADH and FADH2, and it will transfer into the oxygen. That's why it is called aerobic respiration or aerobic metabolism. In this process, it will generate or capture energy. So this is the process of making H2 molecule, but while making H2 we will get energy. So NADH will convert into NAD and give one hydrogen molecule and 2 electrons. FADH2 will convert into FAD and give 2 hydrogen molecule and 2 electrons. Those 2 electrons and hydrogen molecule emerge with each other and half molecule of oxygen, that will give 1 molecule of H2O. So to understand electron transport chain, we have to understand electron transport complexes. As you can see in the picture that there are four complexes, 1, 2, 3 and 4, and electron passes through them and make energy. And how it will make energy, that we are going to see in next slides. So first is complex 1, which is NADH dehydrogenase, which oxidize NADH and will convert NADH into NAD+, and that extracted electron will transfer into coenzyme Q, and that is also called ubiquinone. As you can see in the picture that complex 1, it is doing NADH into the NAD+, and that extract electron will go into coenzyme Q, which is attached to complex 2. So coenzyme Q shuttles electrons to complex 2, which comes from complex 1, which pumps hydrogen into intermembrane space, and key intermediates for those transferring electrons are flavin mononucleotide and iron sulfur compounds. So there are coenzyme Q10 supplements available, which is used in statin therapies. Some data indicates that statin decrease coenzyme Q levels, and which is hypothesized to contribute to statin myopathy. So coenzyme Q10 supplements may help in this theory, but there are no good data to support this use. So next is complex 2. Complex 2 is also called succinate dehydrogenase, and which is also used in TCA cycle. In this, electron from succinate will transfer into FADH2, and those electrons from FADH2 will transfer into coenzyme Q. As you can see in the picture that complex 2, which is succinate dehydrogenase, also used in TCA cycle and also used in electron transport chain. So third complex is also called cytochrome BC1 complex, which transfer electrons from coenzyme Q into the cytochrome, and that pumps hydrogen molecules to intermembrane space. As you can see in the picture that coenzyme Q's electron is being transferred into the complex 3, and in this process hydrogen molecule is going out into the intermembrane space. So cytochromes are a class of protein which contains heme group. Heme group is iron plus porphyrin ring. In hemoglobin, iron is found in mostly Fe plus 2 state, but in cytochrome, iron found in Fe plus 2 or Fe plus 3 state, because it will go into Fe plus 2 and Fe plus 3 state back and forth. So this is favorable for oxidation state changes with electron transport. So in electron transport, there are three types of cytochrome, which is A, B and C is used. In cytochrome P450 is used in drug metabolism in liver. So complex 4 is made up of cytochrome A and A3, which contains copper in it. Cytochrome C oxidase will react with oxygen, and it will emerge with electron and make one water molecule, and it will also pump hydrogen molecule out of into the intermembrane space. As you can see in the picture that those complex will transfer hydrogen molecule into the intermembrane space. As you can see in the picture that complex 1, 3 and 4 will transfer hydrogen molecule into the intermembrane space. That's why there will be high concentration of hydrogen molecule into the intermembrane space. Just know that now. So in phosphorylation, there are two ways that ATP can produce. First is substrate level phosphorylation, and second is oxidative phosphorylation. So in substrate level phosphorylation is going to be with enzyme. As you can see in the picture that phosphoenol pyruvate will convert into pyruvate with the help of pyruvate kinase. So in this process it will convert ADP into ATP. So that's how we will get one molecule of ATP with the substrate level phosphorylation via enzyme. So in oxidative phosphorylation in electron transport chain, there is electron complexes which will throw out hydrogen molecule into the intermembrane space. Because of that, hydrogen concentration will get high into the intermembrane space. I have told you before in the slides. So those higher concentration hydrogen will will make their way into the mitochondrial matrix with the help of ATP synthase, and that will go into the mitochondrial matrix with the help of ATP synthase, and that will make ATP in the way that they will convert ADP molecule into the ATP molecule, and that's how we will make energy via oxidative phosphorylation. So ATP synthase is also called complex 5, and which will convert proton gradient into the ATP, and which will also called electrochemical gradient and proton motive force. So protons moves downgrade gradient, and which is also called chemiosmosis. In this process it will generate ATP. So we'll see how much ATP we will get from each molecule of O2. Classically had to be an integer. There will be 3 ATP from one NADH molecule and 2 ATP from one FADH2 molecule. But newer estimate says that 2.5 ATP from NADH molecule and 1.2 ATP per FADH molecule. So in aerobic metabolism, from glucose we will get 2 ATP, 2 pyruvate and 2 NADH into cytosol outside of the mitochondria, and those 2 NADH will go into mitochondria with the help of Malate shuttle and with the help of glycerol 3 phosphate shuttle. If NADH will go into mitochondria with the Malate shuttle, it will give us 6 ATP, and if it will go into mitochondria with the glycerol 3 phosphate shuttle, it will give us 4 ATP, because NADH is being converted into FADH2 in glycerol 3 phosphate shuttle like we have seen before. So in mitochondria, pyruvate will go into TCA cycle, and that TCA cycle will give us 2-acetyl coenzyme A. So 2-acetyl coenzyme A will give us 2 GTP, 6 NADH molecule and 2 FADH molecule. So 1 GTP is equal to 1 ATP. Therefore we will get 24 ATPs. It means there will be around 30 to 32 ATPs from one glucose in aerobic metabolism. So there are drugs and poison can disrupt electron transport chain. There are two ways to disrupt oxidative phosphorylation. First is block or inhibit the electron transport chain, and second is allow H+ to leak out inner membrane space, and there is called also uncoupling of the electron transport or oxidative phosphorylation, because if hydrogen will leak out of the electron inner membrane space, there will be low concentration of hydrogen and it will not create the gradient that will need for ATP formation. So inhibitors are rotenone, which is insecticides, which binds with complex 1 and prevent electron transfer to coenzyme Q. Entomycin A, which is antibiotic, which will bind to complex 3 or BC1 complex, and therefore it will stops the electron transfer. In complex 4 there are two molecule, which is carbon monoxide, which binds with A3 in Fe+2 state and which will compete with O2. In cyanide, that will bind with A3 and Fe3+ state and it will stops the electron transport. In cyanide poisoning, we will see variety of symptoms. In central nervous system, we will see headache and confusion. In cardiovascular system, we will see initial tachycardia and hypertension. In respiratory, we will see initial tachypnea, bright red venous blood which increase O2 content, almond smell, and anaerobic metabolism which will lead to lactic acidosis, and those are variety of symptoms we will see in cyanide poisoning. So the drug nitroprusside, which is a treatment of hypertensive emergency, also can cause cyanide poisoning because it contains 5 cyanide groups per molecule, and toxicity occurs when there is a prolonged infusion of nitroprusside, and treatment of that is nitrate, also called amyl nitrate, which will convert Fe+2 into Fe+3 in hemoglobin. Fe+3 in hemoglobin cyanide will protect mitochondria. So the uncoupling agents will uncouple the membrane and that will leak hydrogen ion into the matrix, and therefore there will be low concentration of hydrogen into the intermembrane space. So therefore there will be low gradient of hydrogen into the ATP synthase, so they cannot make ATP. And on top of that, those agents will generate heat instead of the ATP. So the uncoupling agents effect we will see in 2,4-dinitrophenol or DNP. Also we will see in overdose of aspirin, and also we will see in brown fat use, especially in newborns or also in hibernating animals. In brown fat we will see uncoupling protein 1, which is UCP1 or thermogenin, which will stimulate the sympathetic system and will lead to lipolysis, and it will, in electron transport chain, it will generate heat instead of the ATP. So oligomycin A, which is macrolide antibiotic, which will inhibit the ATP synthase itself. So proton cannot move through enzyme and proton trapped into intermembrane enzyme. So oxidative phosphorylation stops and ATP cannot be generated. Thank you everyone for watching this video. If you like this video, hit the like button, share it with your friends and subscribe to my channel.